I am so honored to be able to be here today and serve as your moderator. And I'm going to keep my few remarks very, very brief because we have a wonderful keynote speaker. We have a fantastic group of patients who are bravely sharing their stories today, and a fantastic group of clinicians as well to talk about everything. And really, this is all about you. All of you in this room today. It's about sharing, it's about healing, it's about learning. And I want you to keep all of that in mind, and I want all of you to know that to take notes today, if you have a topic that you want to bring up, if you have a question that you want to bring up, whether it's during Dr. Sequist's remarks or something the panelists have to say, just make a note of it, and we're going to try and get to as many of your topics and questions as we possibly can. This is going to be an engaging morning, and I don't want you to just kind of sit there and take it all in unless that's what you want to do. I want you to participate so that you can really get the most out of today as much as possible. So we're going to begin with a wonderful oncologist today, Dr. Lecia Sequist. She is the Associate Professor of Medicine at Harvard Medical School, and her research at the Center for Thoracic Oncology focuses on studying lung cancers and tumor cells and circulating in the bloodstream. And today we're going to learn from her how cancers are being genotyped and what that means for personalized therapy. And that's the key, personalized therapy. So without further ado, please give a warm welcome to Dr. Lecia Sequist. Thank you.
Oh, thank you. It's really wonderful to be here, and thank you to the organizers of the conference for inviting me. It's a thrill to get to talk to patients. Too often I'm standing at podiums giving talks to other doctors, and that can be exciting, and we share our research and really build off each other, but there's nothing like talking to a group of survivors, so I'm very pleased to be here. I'm a lung cancer specialist, and I'm going to be talking mostly about lung cancer today, but I do hope that what I talk about is able to be generalized for you. And it's true that what I'm going to talk about is just one example of how our changing technology and our changing understanding of the biology of cancer is really affecting treatment advances for all different types of cancer. But I will tell you, I never waste an opportunity at a microphone to kind of raise people's awareness about lung cancer a little bit, because I think people don't talk about lung cancer in public forum enough. And so I just want to show you these somewhat startling figures. The reason why I think it's very important to talk about lung cancer and raise awareness about lung cancer is because of the amazing funding gap. So the people on this the size of the little figurines is the relative number of people, Americans, that unfortunately pass away from each of these cancers every year. And you can see that lung cancer is the giant in the room. The dollar signs that are next to the figures are the number of research dollars that go to research on those cancers for each person that passes away each year. So you can hardly see this little blue dollar sign down there for lung cancer. And from my perspective, one of the reasons why there is such a disparity in funding is because there's a stigma about lung cancer, and people think that it's just this guy that gets lung cancer. But really, if you have lungs, you can get lung cancer, and no one deserves to get lung cancer. And so I want to share with you some of the beautiful faces of our patients here at Mass General. This is just a small number of the group of people that inspires me to do my work every day. So enough about that. Cancer is a disease that's caused by abnormal DNA. And what I really want to give to you guys today is almost a vocabulary lesson, because I'm sure that for those of you who are patients or family members accompanying a patient to a doctor's visit, sometimes the doctors will be talking very quickly, and they may use words that you're not familiar with. And so as Liz said, please feel free to raise your hand at any point. I want to make sure everyone understands the words that we're using. So DNA is the building blocks of the genes in all of our cells, the genes that are passed down through, from mother and father to child. It's important to know that not all of the cancer genes that we talk about are hereditary. There's a difference between the types of genes you could pass on or you could have inherited and the types of genes that are in the cancer cells. Because when you're already an adult and you have a set of genes, sometimes something external can damage the genes that are there, and that can lead to cancer. And sometimes, in some cases, cancer does come from a gene that you inherited. So there are different kinds of genes. But basically, cancer is a disease where one of the genes or the DNA has something wrong with it. And what DNA does is it makes a protein. So I wonder if we have, just use this as a pointer. So the genes are here. This is the DNA, the kind of double helix. And what a gene does in a cell is it tells the cell how to go about its daily business. And the main way that cells do all their business, the building and the growing and the metabolizing, is through proteins. And here in this cartoon, the protein is this thing that looks like a football goal or an antenna on the surface of the cell. So this is supposed to be a normal cell with normal DNA and a normal protein. And then a cancer has something wrong in the DNA that makes an aberrant or abnormal protein. And what these proteins do in this case is they send signals to the rest of the cell. And so a cancer protein that's abnormal may be sending abnormal signals, such as signals to grow more than normal, signals to spread to another part of the body. These are the kinds of things that cancer cells do. And so one of the breakthroughs in cancer that we talk about a lot is targeted therapy or personalized therapy. What does that mean? Well, this prior graph was very simplistic, right? We can understand this antenna sending signals. This is a little bit of a tongue-in-cheek. When I show this to doctors, they usually laugh because the idea of this picture is that if we could understand all the inner workings of a cell and know that this particular area connected to this and communicated with that, then we could draw a diagram of it. But the reason why it's tongue-in-cheek is that we really don't even have the understanding yet to all of the inner workings of the biology of a cell. But this is actually a picture that's nearly 10 years old now, trying to depict all the connections between what we knew about biology at that time. And the idea with targeted cancer therapy is that if you can understand the inner workings of a cancer cell to this degree, you can then, in an intelligent way, block one of these signaling pathways and shut down the way the whole cancer cell functions. That's the idea of targeted therapy, finding that Achilles heel or that most important point where a blockade shuts down the whole system and there can be no backup system that goes around the blockade. So that's the idea of personalized medicine in cancer or targeted therapy. The other analogy I use with our trainees, our fellows who are learning to become oncologists, is this analogy of the vault door and the lockbox key. So you know that if you have something valuable that you're going to put in the bank in your safety deposit box, if you then want to get it out later, there's two basic steps. You have to have someone, usually the bank, open up the vault door, which is this giant thing with lots of moving parts and big steel tumblers. But that's not enough. Once you get in the vault, you still have to have your key that opens your particular box. So the analogy for cancer therapy is that understanding that biology, understanding that complicated diagram that I showed you on the last slide, that's opening the vault door. So if you have a particular cancer with a certain situation, like a gene that's driving its growth, having all the scientists work to unlock and understand the diagram of how it works, that's opening the vault door. That's not enough to treat that cancer. In order to get that cancer to die off, you have to have the key to the lockbox, which in this analogy is a drug that works in the context of that particular cancer's biology, a drug that is actually able to be taken by people without, crippling side effects. So you have to have a drug that works for the scenario in order to get to your gold or your pearls or whatever you have in your lockbox, in this case, cancer shrinkage. And I'll show you some pictures of patients' scans and how that can work. So that's really what targeted therapy is all about. And then in the clinic, the idea is that you can take a group of patients in the center of the slide here who historically would have all been treated the same. For example, in my field, these would all be patients with adenocarcinoma of the lung. We used to treat every patient who was diagnosed with adenocarcinoma of the lung the exact same way, with a series of different chemotherapies in kind of a cookbook fashion. But now we can use some of our technologies to really parse them out into different groups of patients that maybe have different driver mutations and should be treated with different algorithms that are more specific to the biology of their cancer. So that's what this idea of genotype-directed therapy is all about. And in lung cancer, for example, this is a pie chart of adenocarcinoma of the lung, which is as recently as 6 or 7 years ago was really thought of as one disease, one monolithic group of patients. Now it's very widely appreciated that it's not one disease and that there are different driver mutations, changes in the genes that are causing a specific abnormal protein and signaling from that protein that should be treated differently. And in lung cancer, the real breakthrough, I think, is that not only have we understood this, but we've also been fortunate enough to have drugs being developed at the same time that happen to fit these different pieces of the pie so that patients can be treated differently. So the first area where we really had success with this concept was with patients who have a mutation in EGFR. And these patients have been very well treated with a pill that inhibits EGFR. Then, and the EGFR mutation was actually discovered here at Mass General, and the breakthrough about treating patients with ALK translocation also happened at Mass General. So, we've been very fortunate to be a key piece of the research moving this type of treatment forward. So now there's an FDA-approved treatment for this group of lung cancer patients that has ALK translocations. That treatment also happens to work well on this smaller piece of the pie over here, MET amplification, and this smaller piece of the pie, ROS translocation. So some of these drugs, just by luck, may work for more than one piece of the pie. But the important concept is that each piece of the pie is really a different type of cancer. Now, something else that's exciting is that some of these pieces of the pie are not just a newly appreciated type of lung cancer, but they can also sometimes be found, these different gene abnormalities can be found in other cancers. So this morning, one of the patients that you'll hear from later was commenting that some of the advances in breast cancer have actually influenced patients being treated for stomach cancer. And there are many examples of that as well, where one of the breakthrough treatments for melanoma has really influenced the treatment of patients with lung cancer that have this BRAF mutation. So, historically, cancer doctors have really put their cancer patients into boxes. This is breast cancer box over here and a colon cancer box over here. And I think our increased understanding of how the biology of cancer works has not only split those boxes into many components, but we've realized that there are links between some colon cancers and some lung cancers really should be treated more like each other than maybe another patient with colon cancer. So the whole paradigm of how we think about cancer patients and how we approach treatment decisions has dramatically changed. I think, as Mara said, a tectonic shift. It's really a different world, and it's a very exciting time in cancer research. So here are some CAT scan images of some lung cancer patients who have been treated with these targeted drugs. So a CAT scan is a slice. Think of it as if your body is a piece of bread and we're looking at one slice at a time of the loaf of bread. This is a patient lying on their back, and this is their their right lung, and this is their left lung. The left lung in this picture looks more like a healthy lung should. It should be filled with air, which is black on an X-ray, and it should have little white lines through it, which are the blood vessels and the air sacs within the lungs. So you can see that in this patient, her right lung was really replaced with the lung cancer. She had an EGFR mutation and was treated with this drug called gefitinib or Irissa, and after 6 weeks, you can see that this cancer is really melting away and the air is coming back into that lung. Here's a patient with ALK rearranged lung cancer. Again, you can see all of these spots of cancer in both lungs, and after 12 weeks on the pill crizotinib, those are going away. Here is a patient with Ross rearrangement, and in this case, both of the lungs are involved, but mostly the left, but the right is quite abnormal too. And after 2 months, the lung, this is really what a normal-looking lung should look like. It's really normal after treatment with a targeted drug. And here's a patient with MET amplification. So not to keep being repetitive, but you know, I have to say that these types of responses are just beyond anything that we used to see with chemotherapy for lung cancer. So they're really quite amazing for both the patients and the providers who are seeing them. So this is a patient of mine who had MET amplification and went on crizotinib and had really a complete response to that, which was maintained for quite some time. So the advances so far that we've seen in lung cancer treatment have really been finding these mutations, these kind of single abnormalities that have that have such an impact on the biology of the cell that you really can shut it down with one particular blockade. But not all cancers are going to be that easy to shut down, and even among a group of patients, say with an EGFR mutation, there's a range of the success of these treatments. And some patients are able to stay on the treatment longer, and some patients it doesn't work for as long. And so we're still trying to uncover all of those little factors that play into the variation there. And so this is a diagram that shows some of the different types of genetic analyses that can be done. The genetic analyses that we've talked about so far are really the first pass, the simplest of the ways of looking at the genes. And what we've been looking at so far is here in the middle, what's called a single nucleotide variance, or really a mutation, just where one, you know, the genes are like a book. Okay, so there's letters, there's words, there's paragraphs, and when you put it all together, it's a story. That's the blueprint of how the genes read. And so what we've been looking at basically is one word that's been taken out and replaced by another word, but it's a crucial word so that the whole context of the paragraph and really then the story changes. You can imagine that, you know, those don't happen all that often. And actually back at the pie chart, there's still, in lung cancer, a large chunk of the pie where we don't know what the key mutation is. And I think in that case, it's not that there is one key mutation. We have to start looking beyond just a typo of one word, and we have to look at the whole context of the genes or the genome. So other ways that we as scientists can look at cancer genes and try and figure out what's driving the abnormal growth here. We can look at copy number. Is the cancer making a whole lot of copies of one word so that that word in the story gets overemphasized? We can look at the protein pattern. This is actually what happens downstream of the genes, the proteins that the cancer makes. We can look into the proteins and see how they're being translated from the genes. We can look at gene expression. So when you have this whole book, which is the DNA on all the chromosomes, it turns out that the cell, when it reads the genes, it doesn't read the whole thing. It reads kind of the Cliff Note version. And there are certain signals that tell it what portions to read and what portions to skip over. Those are the genes that are actually expressed. And so cancer can sometimes change the way that the cell reads. It can bring certain paragraphs into the forefront and push others down behind. So that's what gene expression is about, and our technologies now can look at gene expression. We can look at rearrangements in the chromosome. This is a kind of a neat way that they diagram rearrangements where, I don't know, you probably can't read this, but each one of these little black smudges here is actually a number of the chromosomes. You know, we have 26 chromosomes. I don't know why it's like this, but basically these little pink lines that are going across the circle tell you that something from chromosome 2 has been moved over to chromosome 3 accidentally. So these are rearrangements. Epigenetics is also a very interesting area in cancer research, where this is, again, how the cell decides to read part of the genes or not read the other part of the genes. So newer technology is allowing us to look at much more detail about what's going on in the cancer cell. And I think that having these types of tests come into the clinic is going to happen quickly, the way it did with genotyping and looking at the mutations. You know, just 6 or 7 years ago, looking for mutations in cancer was thought to be something that was really in the realm of research. It's something that you would do not in real time as the patient is deciding on treatment A versus treatment B, but something that could be looked at later, and then you'd see all the patients that had treatment A, how did they do if they had this gene. Once we actually got those drugs that matched with the patient genes, that's when we realized we can't do this for research later. We have to do this now while the patient's in the clinic so that we can decide, should they go on treatment A or treatment B? And that led to this revolution of bringing gene testing into real time, into a test that would be ordered by any oncologist in the community and get reported back to you as part of the medical record. Some of these other areas are currently, some of these other analyses that can be done about the genes are currently in that realm of, oh, this is something that will be done for research. We can look back later and see how people did. But I think very soon they're going to be moving right into the clinic, into real-time analyses, just like the genotyping did. And part of the reason why these things are possible is just the amazing explosion in technology. So just like you've all experienced with your phones and your computers and everything else in your life that has gone amazingly wireless and virtual and mobile, that's the same thing in medical research. And so this is, you may remember, over a decade ago when they sequenced the first human genome, it was huge news. It cost thousands and thousands of dollars, and it took almost a decade to complete. And the cost was astronomical. And then there was this scientist named Moore who postulated that as technology advanced, the cost of us looking at a megabase of DNA would go down. And this was, his line was sort of the predicted rate at which that cost would fall as technology advanced. But you can see what's happened in reality is we followed Moore's law for a little while, but then there was an explosion of sort of technological and bioinformatics understanding, and since then things have fallen dramatically. So that whereas it took a worldwide collaboration and literally millions of dollars to sequence the first human genome in a decade of time, now sequencing a genome might be something that a college student would do over a summer research project working in somebody's lab. It's amazing how quickly our capability has changed. What about, now a question that always comes up when I give this kind of talk is, what about patients who don't have a particular mutation identified in their lung cancer at this time? And something else that's been very exciting this last year or 2 years that has affected multiple cancers, such as lung cancer, melanoma, kidney cancer, is this idea of harnessing the immune system. And I think that one of the other changing landscapes that you're going to see in cancer care over the next couple of years is an explosion of understanding and of bringing treatments to the bedside that harness the immune system. The immune system is supposed to be on the lookout for cancer cells in our body, and it is supposed to kill cancer cells if it sees one develop. And actually, most of the time that does happen. But if someone gets clinically diagnosed with cancer, that's a sign that something went wrong in that process, and the immune system wasn't able to wipe out the cancer when it was only 1 or 2 cells, and it was able to grow. And for a long time we've known this. For a long time people have wanted to take the immune system and teach it how to fight cancers. There's been a lot of research into cancer vaccines. Unfortunately, for the most part, these therapies have not worked over the last couple of decades. Last year there was a very exciting vaccine that was approved for prostate cancer. But other than that one example, we haven't seen a lot of success with vaccines. But there's a new type of immune therapy that has been very successful in multiple kinds of cancer. And what that basically does, this is a diagram, this is called the programmed death pathway, the PD-1 pathway. And what this diagram basically shows is a cartoon of how this works. I describe it to my patients a little even more simpler than this cartoon, which is that the immune system is out there looking for cancer cells, but your cancer has somehow evaded the immune system surveillance. And it's done that by putting up a camouflage in front of it or a facade so that the immune system just looks right past it. It doesn't notice it sitting there. And this new type of treatment that targets PD-1, what it basically does is it lifts up that camouflage and that facade, and all of a sudden the immune system sees the cancer sitting there and it starts to attack it. And so this is an early treatment, but it's definitely been very exciting in the field of lung cancer, as well as melanoma and kidney cancer, as I mentioned. So that's another thing to keep your eye out on, I think, as we look at the changing landscape of cancer care. So the take-home points that I want to leave you with is that targeted therapy really has become a reality. Today I focused on lung cancer, that's my field of research, but it has become a reality in many different types of cancer treatment paradigms. And I think it's here to stay. Biomarker detection technology is advancing at light speed. So I showed you about Moore's Law and all those different types of new analyses that are going to, I think, come to the bedside very soon. The limit is no longer what can we do, it's do we have the bioinformatics to interpret all of the data that's generated from this. So if you have children or grandchildren who are trying to figure out what to do with their life, tell them to go into bioinformatics. It is going to be the biggest demand. It already is a giant demand to have smart young people who can deal with all of the information and pull out the pieces that we need. Other types of novel therapies are proving successful, such as those that harness the body's own immune system. And I would say there's never been a more exciting time in cancer research. So I'm glad that you're all here today. I hope that we're going to talk more about some of the exciting advances in cancer, but also, how it's affected you in your own life and from your perspective, what you see with these types of scientific advances. So thank you for your attention.